Welcome everyone. My name is Lawson Allen Jr. I'm a senior pre-dental chemistry major at Georgia Southern University. Today I'm going to be talking about atomic force microscopy, or AFM, for my Chemistry 4241 instrumental analysis class. Right here I have a picture of an atomic force microscope. As you can see, it has many features that are familiar with other microscopes, but there's also some added in features that make it a specific technique, and we'll go into that later in this video. A scanning probe microscopy refers to a group of imaging techniques that capture an image of a sample surface. The atomic force microscopy technique is the most commonly used scanning probe microscopy technique. It's used all over the world, and it's used for pretty much any skill level, from undergrad all the way to researchers. For AFM, almost anything that is solid can be used as a sample. This includes polymers, metals, hair fibers, nucleic acids, proteins, minerals, lipid bilayers, which is what we used in this research article that I'll explain later. It can be used on cells, viruses, bacteria, plant surfaces, anything like that, etc., can be used for the AFM. Some advantages is it can be used in a vacuum, it can be used in air or a liquid. A liquid is usually, however, required, and that liquid usually uses water or a buffer. The AFM is a type of surface profiler. It gives an image to the fraction of a nanometer of the surface of the sample, so we have a very finite picture of what we're looking at. The AFM collects images of a sample surface by moving the probe over the surface in a rastern pattern, which means the sample moves left to right under the probe as it goes back and collects this image. As the sample moves, the probe gets pushed up and down, and it moves across the surface, which allows the probe to measure the height and therefore create an image. There's a tip that interacts with this sample that I'll show you in a picture in just a minute. A laser is shot on the probe, and the angle that is reflected off the probe is detected and then collected by a feedback method. It's a very specific mechanism that involves a probe, and a probe consists of a cantilever and a tip, but I'll also show you that in a coming slide. An image is finally created by plotting its horizontal and vertical movements as the tip has scanned across the surface. We get a feel for how the surface looks by how much this tip has been affected backwards and forwards and up and down. Right here is a basic layout of the atomic force microscopy. You've got your laser beam that shoots a laser onto a cantilever. This cantilever holds a tip, and as the tip goes across the sample's surface, the cantilever, based on the height and movement of the tip, the cantilever changes its angle. That angle changes the laser's angle that bounces off, and then we have a photodiode detector that reads and picks up this angle. Also, I've called it a probe. A probe just simply means the cantilever and the tip. It's referred to as the probe. As we discussed, the major components of the AFM are the laser, the photodiode detector, the probe, and the sample. The laser is used to measure the deflections caused by the adjustments of the probe due to the sample's topography, or the height of the sample. The photodiode detector detects the changing laser angles and receives the different changing angles from the laser, and helps develop our image from that. The probe consists of the cantilever, as I've said, and the cantilever holds the tip, and the laser reflects off the cantilever. The tip fills the surface of the sample and moves horizontally and vertically to analyze the topography of the sample surface as it moves across in that rastern pattern that we talked about. And then, of course, you have your sample, which is the analyte that's being analyzed or determined. In AFM, there's signals or the data that you collect. The data that is collected from the AFM is an image of a sample surface. That's the main point we want to get across. We want this really high resolution image of this surface. This image is obtained by using a laser and a probe, like we've talked about, to analyze the topography of the sample and then plotting it to create a high resolution image that can be viewed. The qualitative data we receive is the adjustment of the tip to keep constant laser deflection. Each analyte's going to have a different one, so we can figure out different analytes by that. And then the quantitative data is obviously the intensity of the laser adjustment, how strongly it has to move. The research article I did was called Visualizing Water Molecule Distribution by Atomic Force Microscopy. Those are your authors. Their sample was a muscovite mica substrate from muscovite mica water systems in the body. Samples were studied by the cross-sectional of liquid hydration water layers in the Y direction against the solid muscovite mica surface in the X direction. And this is how they plotted their picture. Their goal was to visualize the two-dimensional cross-sectional hydration structures on that very high-resolution atomic scale. Here's some of their results. Each graph shows the force plotted against the distance of the tip that was moved on the surface, and the picture under it is what actually was received from the different laser deflections that they received in their photodiode. Using the AFM, the image of the muscovite mica substrate and hydration layer were created, as we just saw in that picture. The two graphs showed the force versus the distance. The force is created between interactions of the tip and substrate complex, and the distance is a deviation the tip occurs based on the topography of the sample, like we talked about. And then, of course, here's the references. I appreciate you listening. I hope this has been enjoyable and you've learned something from it. And have a great day. Thank you.